So a video covering the part of the syllabus on brain scans and the key study of Raine. So remember, we've got various methods of scanning the brain, positron emission tomography, PET scans, which study the activity of the brain, and magnetic resonance imaging, which studies the structure of the brain. So remember, a PET scan allows us to see the activity of the brain. What we do is we inject you with a radioactive chemical, usually glucose of some kind, and as it breaks down, as your brain uses the chemical, it breaks it down, and so the parts of your brain which are active, the parts of your brain that are really working, will use up more of the chemical, and so show up in the brain scan as being red. Red, yellow, things like that. The parts of your brain which are not as active will show up as darker colours, and the blues and the greens, things like that. So we can see your brain being more active, the more radiation it uses, the brighter it glows. Remember, so we use PET scans to look at the activity of the brain, and this has strengths and weaknesses. It is harmful to people to give them this injection. They might have claustrophobia. However, it is reliable, it is scientific. It does it's an objective measure, although it can be hard to pinpoint exact areas of the brain rather than just areas that are in general use. For example, if you're lying there and we're trying to get you to do maths, not only the part of your brain that does maths might light up, but also the part of your brain that's sensing the sensation of lying down, things like that. So it can be hard to pick apart different areas of the brain. MRIs is the next one. Remember, MRIs show you the structure of the brain. What we do is you line a large cylinder and we bombard your body with radio waves, and you're in like a big magnet. That magnet bends the radio waves, and it affects the atoms in your body. The computer is able then to pick up how those atoms are affected. By doing so, we're able to see the structure of the brain. So MRIs show you the structure of the brain, so we can look at things like damage or where there's a tumour, things like that. Remember, they also have the same strength. They are scientific. They are less invasive, although you can inject people with a tracer, and they are extremely claustrophobic. One of the big weaknesses, though, is it doesn't measure activity. It just measures structure. So Raine 1997 is one of your core studies. Remember, the aim of this was to see if the brains of murderers were different to the brains of non-murderers in some key areas like the prefrontal cortex. So Raine got 41 murderers, 39 males, 2 females, with an average age of 34.3. They'd been charged with manslaughter, and all were found NGRI, not guilty via reasons of insanity. Some had brain injuries, some had drug abuse, some had a history of epilepsy, some had a personality disorder, things like that. And what we did was we compared them to a control group. This was 41 normal individuals, non-murderers, matched for sex and age, matched on other things like whether they had schizophrenia. So there was 6 with schizophrenia in each of the conditions. Some of the things we couldn't control for were handedness and race and head injury. These were things that might have affected the results because they could cause differences in the brain. Being left-handed rather than right-handed could cause your brain to be slightly different. Having a head injury causes your brain to be different. So that was something that we couldn't control for. So it's a natural experiment. Remember, whether you are a murderer or non-murderer is an IV that we are not in control of. So it's a natural experiment, and it was a matched pairs design because of those pairings that we did. It takes place in a lab. So the procedure, each participant carried out a practice test, and then we injected them with a tracer. FDG is just the acronym for that tracer. The participants then completed a target recognition task. So they had to identify a shape in amongst a field of other ones. This was to get their brains working and get that tracer being used. After about 32 minutes, they were given a PET scan. So they were put into the tube, and we looked at the activity of their brain. There was 2 methods we used: the cortical peel technique, where we looked at strips of the brain, and the box technique, where we looked at 2 centimetre squares. Both of these are designed so that we can look at the activity in the brain. The results, there was no difference on the working task, so they all identified the shapes equally as well. We see difference in some of the cortical regions. So the prefrontal lobe had lower glucose metabolism in murderers. Remember, the prefrontal cortex or the prefrontal lobe is responsible for things like decision making and control of behaviour. So the fact that murderers had the lower activity in this would indicate that they were less in control. The parietal lobe also had lower glucose. The temporal lobe showed no difference. The occipital lobe had a higher glucose metabolism. They had lower activity in the corpus callosum. The amygdala had reduced activity in the left and more activity in the right. Remember, amygdala is related to emotions, so this is why possibly the murderers might be more aggressive, because they have more activity in the right amygdala, which is tied to aggressiveness. We know this because we've done studies on cats where we stimulate this area of the brain and they go into kind of a rage. And then we see similar patterns in the medial temporal lobe and the thalamus. Handedness only had a slight impact, race had no effect, and so on. So in conclusion, those who are NGRI have reduced glucose metabolism in the prefrontal cortex, posterior parietal cortex and the corpus callosum. They also have abnormal activity in the amygdala, thalamus and the medial temporal gyrus. These are kind of key bits of finding. So the prefrontal deficits explain the lack of self-control. They can't control their behaviours, which is why they kill people. Posterior parietal cortex, I'm saying why they kill people. Obviously, we can't derive cause and effect from this, but this is one explanation that people have given. The posterior parietal cortex could result in reduced cognitive functioning, so they have less verbal ability. They are less able to express themselves or understand others, which might have affected their level of schooling. The corpus callosum, their brain is less able to communicate with each other, so the left hemisphere, which is normally in control, is less able to regulate the right hemisphere. And finally, kind of a key bit, the amygdala explains why they're more aggressive. Remember, we have to be able to evaluate this study, and to do so we would use Grave. We'll do that in class though. Thanks for watching the video.